So first of all, there's more of a bio online if you want. I'm a medical doctor, and all the slides are online, and there's hours of video on YouTube, so don't feel like you have to take screenshots and stuff. Everything's publicly available. But first, I'm a medical doctor, usually presenting to audiences. I got to talk about my conflicts of interest, so I just have a few. Suffice to say, I've started a bunch of companies. I consult for a bunch of companies. You probably don't want to believe another word I say over the next 20 minutes, wouldn't blame you. I'm in professor mode already with my stick here, by the way. But I'm most proud of the bottom right. Those are all the companies started by my students. More than half my graduate students start companies now. They do this even if they become academicians, professors. They should still start a company anyway. And they do this with the best platform in the world. And you know what that is. That's data. It's often big data. It's often open big data. So I'm going to talk a lot about open data here, open health data. So I'm going to show you how they do it. I'm going to show you how I do it. Maybe I'm going to convince you this is the most amazing time to be in health data sciences right now. It's just an unbelievable time. We're in this amazing mode now of trying to get to precision medicine. So President Obama himself mentioned precision medicine in the State of the Union address a year ago. And what that really means is we're going to be able to, we're entering this era where we're customizing the healthcare we deliver to a person based on the measurements we're making on that person, right? It could be molecular measurements, DNA, it could be your Fitbit, it could be wearables, but we also have to take into account all the measurements we're making on everyone else, right? It's not just you, but we got to learn from everyone else that we've been taking care of and then apply it to you. So we got to record what we're doing to all the patients out there. It's public health, but just bring it into you. And you can see the three reasons why the government thinks it's the right time. It's sequencing of the genome, which is data rich, biomedical analysis, data rich, and new techniques for using large data sets. And they literally show a picture of the cloud. I mean, the three and only three reasons the federal government wants to do this is because we're in this data era, right? I mean, you can't get better than the president of the United States pushing for this, right? So we're in this data delusion healthcare because of amazing gadgets like this. Now I could have showed anything. I could have showed you Moore's law plots. I could have showed you a sequencer. I love showing these because this is how I got my start in the field with these little kind of nifty gadgets. You put any sample on there. You got to process it first, but you put a cancer sample, diabetes sample, mouse model if you're into that, and it gets a readout of every gene in the genome, like how much of each recipe is being made. A lot of this one, not so much of that one. Now when I got my start in 1998, 1999, these were priceless. We had 60 of them, 60 kind of cancers. We were analyzing that. And now they're so cheap, I carry one in every suit just for talks, okay? It's also the thing is, big data is kind of ethereal, so I like to show big data comes from small packages, just like this, right? In fact, now everyone runs these chips, okay? It's not even a big deal. In fact, when you get tired of measuring them one by one, you measure them in the 96-well plate format. And that's so cheap, I have that in my bag. This one's kind of falling apart a little bit, but pretend you see 96 squares there, right? And then when you get tired of measuring them 96 at a time, we now have the 384 well plate format. And that's so cheap, I can put that in my bag. I cannot more clearly illustrate exponential growth in biomedical data than to show you these plates, right? I started with one by one, and now we don't even use these anymore because we moved on to sequencers. In fact, this company doesn't even exist anymore. They just got acquired, Affymetrix, 3 months ago. That's how fast this field moves. In fact, so many people use these chips that everyone started to write a scientific paper, a publication, right? Here's a list of genes that change in that disease, and this disease, and heart failure, and cancer. And they all started to write their papers and submit them to journals. And what did the scientific journals say? They said, hold on a second, we're getting too many of these papers. No more papers with these chips in them, no more, okay? Unless you put this data into an international repository where the reviewers can double check the math. It's probably important to do that. We're getting to patients now. And maybe others can use these samples for something else. So this precedent in science goes back almost 38 years. We have something called GenBank. You may never have used GenBank. You might have heard of GenBank. It came with the first DNA sequencing papers. There's a figure 1, it has A, G, T, C, these letters of the DNA. Nobody wants to type that in again, right? So they're sending tapes to each other, right? This is even older than 8-inch discs, if you remember those, which are older than 5-inch discs, 3.5-inch discs, CD-ROMs, DVDs, and of course the World Wide Web. So for 38 years, scientists have been sharing raw data, and the precedent is there. If you get enough scientists to use the technology, you got to share the data eventually. Fast forward to August of 2012. This article came out in Nature, one of the magazines. What happened in August of 2012? We hit 1 million samples publicly available. 1 million cancers, 1 million diabetes, 1 million biopsies. Someone already got the sample. Someone already processed it. Someone already digitized it. Data freely available on those samples. Now this is de-identified data. You cannot figure out who these people are. but you can get an idea of what that sample is doing, what that tissue is doing, what that disease is doing. You can see the growth curve from 0 to 1,000,000. In fact, if you look really closely, it looks sad because it looks like it's slowing down there at the end. And then you realize they only counted half the year. That was August of 2012, okay? Still doubling every 4 years. We're just a couple thousand short now of 2 million publicly available. Probably by the end of this month or next month, we'll hit 2 million samples publicly available. So what does that mean, right? Any of the scientists at UCSF or Stanford or anyone, any good scientist can go now, instead of starting at the bench, at the wet bench with a pipetter, they can start looking for data, right, on cancer or diabetes. Forget our scientists. Even a high school kid now that needs to do a science fair project, she can go to these websites. For example, if she needs to do a science fair project on breast cancer, type in breast cancer, hit search, and find and download nearly 70,000 samples of breast cancer for her science fair project, about as easily as she can find a song on iTunes today, okay? 70,000 samples of breast cancer already ready. But what's so magical about that number, 70,000? If you haven't figured it out, that's more samples of breast cancer in the repository than any one breast cancer researcher will ever have in their lab. Because every one of them eventually has to send data into the middle. And whoever figures out the middle now has more data than actually any scientist in the field. And if it's not breast cancer you like, maybe it's colon cancer, prostate cancer, they're all there waiting for you. Because can I tell you, these scientists, when they get their grant money and they go make their measurements and they go write their papers, what do they do next? Yeah, they submit the data and then they go get another grant to go get more money to go get more samples done. Nobody in this field in general knows how to use this data and just sits there doubling and nobody's using this data. So I'll give you 2 anecdotes of what we did with this kind of data. The first is the first big grant that I got when I was still at Stanford was go get every human disease studied by these chips that we public data for. Cancer, diabetes, not just cancer, what kind of cancer? We started to collect experiments of disease and healthy. And all that data is in the repositories. We use text mining to figure out which disease was which. We human eyeball it to make sure we got the right diseases in the right place. And you know, I'm not even going to show you genes and chemicals and molecules. I'll show you these pretty little boxes. The boxes, each one is a gene or a recipe in that tissue. And the colors maybe mean more of this or less of that. And we calculate these signatures. So signatures means what's changing in that disease from healthy to normal, right? What's the difference there? So what are we going to do with this? Well, if we got an idea of what's going on in the disease at this level, maybe we can use that to make a blood test, a diagnostic. What's a diagnostic? It helps us tell, as doctors, what kind of disease a patient has, right? What do they have in front? You might have imaging diagnostics like an X-ray or blood tests. And so we wanted to use this data to help us make blood tests. And so we did a lot of these, but the one I'll spend a little bit of time talking about is this disease called preeclampsia. Any of you have heard of this disease, preeclampsia? Show of hands. Yeah, so it's usually about a third to half the audience. It's a half that actually watches Downton Abbey because one of the characters dies of this disease. Spoiler alert, if you haven't watched it. Actually, that dies of eclampsia, right? So you can be pregnant, you have high blood pressure, you start seizing, that's kind of a nasty thing to happen. Preeclampsia happens even before that. It's still how husbands often lose wives around the world, okay? It's a nasty disease. And believe it or not, our diagnostic for this is the most non-specific thing in the world. We just look to see if there's any protein in the urine. It's just so crude, and still, this disease just keeps happening. And a lot of people might have friends or family that have had this happen. There's no good diagnostic for this. So we wanted to come up with diagnostics. So there's 3 names at the bottom there. Linda Liu is a grad student. Bruce did the protein work you'll see in a second, and Matt got us involved at the end here. And so the idea here is let's just search the repositories for preeclampsia. Okay, so let's just search. Here's 266 experiments here. Here's one on preterm birth, preeclampsia, prematurity. How many more do you need to get started here? Someone's already studied this for you. They've run got the samples, ran the chips, deposited the data. And so what's the methodology here? I'm not going to show you any code. I'm not going to show you any formulas. Just imagine a massive Venn diagram, right? I'm trying to figure out what's in common across all these experiments. What are they all seeing? I don't trust any one of these experiments, but I trust what they get in common, right? That's wisdom of the crowd here. I don't trust one of them. I trust what they all see in common. So we chase down what they all see in common, and we start to do some protein work. And here's an example of a new protein blood test that works. So it's called HPX, hemopexin. The pink are the preeclampsia women. The green are the non-preeclampsia women. And you can just by eye see that it's higher, just as we predicted here. We found like 7 or 8 of these. We filed patents. We wrote papers. And what do you do next in Silicon Valley? You start a company on this, okay? So you got the unmet need, you got the data. We had March of Dimes funding the early work. We analyzed the data, designed the diagnostic. We got one of these seed grants at Stanford called Spark. We have them at UCSF called Catalyst. They give out $50,000 if you have a good idea. We got the test to work with the samples in the repository, and then spun out a company called Carmenta, raised $2 million seed financing. Now that's an interesting story. This was a one-man company, Matt Cooper. We launched a multi-center trial because you got to test this forward now prospectively. And why do I start with this one? Because I love this story. We already sold the company. It's already been acquired. So we went from public data, all right, free data. You don't even need a user ID and password to get to this data. Designed a diagnostic, tested it in the lab, launched the company, got seed financing, sold the company 24 months, start to finish. Inventors happy, investors happy, and we're going to do more of these. You bet we're going to do more of these. I'm giving away my secrets, guys, because we need so many diagnostics in medicine. You could all pick a disease, and we'd never step on each other's toes, okay? So that's a diagnostic story, from zero value public data to an acquired company in 24 months. Second story is on drugs and pharmaceuticals. The story is much worse there. You know, if we think we don't have diagnostics, we don't have enough therapies, therapeutics, because everyone thinks it costs $1 billion to develop a new drug. It's actually, that's an underestimate. It's not $1 billion. It costs way more to develop a drug. You know, by the way, how do you get the right number of how much it costs to make a drug? How much did you spend, divide by how many drugs did you get, right? Simple math. What did you spend, divide by the number of drugs? And for these top 12 companies, it's between $4 billion to $12 billion per drug. Kind of not sustainable, okay? You may love the pharma industry, you may hate the pharma industry. Look, I consult for them all. I love them still. But I don't have faith that they're going to solve this problem at all, okay? It's just not even the right culture. Forget about picking on the pharma industry for a second. Even if every pharma and biotech company on the planet was successful, there are not enough of them to develop all the drugs we need for this modern era of precision medicine. So then you realize it has to come down to us to solve this problem, typically in Silicon Valley. Like, we cannot keep it going at $12 billion. We've got to figure out cheaper ways to develop drugs. So how we're going to do this, we're going to use public data. That's my approach to do this. And you know, while we're turning our crank, we started to realize if we can collect the disease and healthy on the left, we can start to mine the experimental data for cells treated with and without drugs. Here are some cells in a petri dish with the drug and without a drug. And they run the same kind of chips to get the same kind of measurements, and we're going to put these two together. The reporters for this call this Match.com for drugs, okay? You know, Match.com. And what's a famous saying when you're looking for a spouse or a mate, right? Opposites attract. So here's my informatics methodology. If I've got a disease on the left where this gene goes up and this gene goes down, and I can find a drug that can make this one go down and this one go up, maybe there's a match there. It's as stupidly naive as that, okay? I'm just looking to reverse the profiles. That's 2 genes, imagine 20,000 genes. You can imagine Kolmogorov, Smirnov tests and all that stuff, right? So we turn our crank, we got lots of ideas. You know, here's a drug, there's a drug, but actually where we get a lot of traction is here's a new use for an old drug, this idea of drug repositioning. Because these are drugs that we already know the safety profile for, but maybe there's something else that they're actually good for. So we turn our crank, we get lots of those. And this, we've done more than a dozen. This is my favorite. This is on publications. All this stuff is open access. You can get to it. Here's a drug called imipramine that we use for depression. Okay, it's called a tricyclic antidepressant. We used to use it for depression. We actually don't anymore. We use Prozac, these serotonin reuptake inhibitors. And this is a mouse that has lung cancer. You can kind of see the lung nodules here. You knock out these genes, the mouse gets mouse lung cancer here. And on the right is the same kind of mouse with the lungs, all better with the imipramine. Now, we don't use imipramine anymore because it's got some side effects. It makes people sleepy, and it might set you up for an arrhythmia. It might set your heart rhythm a little bit off, causing a little bit of a disturbance in your heart rhythm. Actually, neither of those 2 side effects sound as bad as having lung cancer, 5% survival rate. Only 5% of people survive after this diagnosis in 5 years. And the cancer is melted away here. It's gone in this mouse. Now, why do I love this one, okay? This one's amazing because we start with public data. We get the prediction this drug is going to work on lung cancer. We test cell lines in a petri dish. I'm not even going to show you that. I got this lung picture. 15 months after the computer prediction, we got the ethics board approval. We launched a clinical trial on this one. Humans are dosed with this drug now. This is a computationally driven clinical trial. 20 patients recruited on the study. Total cost of this trial, $50,000 bucks, okay? Not even $1 million, not even $100,000 bucks. $50,000 gets us this trial going. We got to do way more of these, okay? And not spend $1 million or $1 billion to do it. We got to get way more of these to happen. No pharmaceutical biotech is targeting small cell lung cancer today. There's just zero. None of them are. There's just too many other diseases they're working on. And what do? You got to start a company. So this one is NewMedi. We've raised more than $5 million on this one. And I love this story. So of course, my wife runs this one, so I love this one the best out of all my companies. Gotta love this one. But this little team of 7 people over in Palo Alto, kind of like in a garage. I call these garage biotechs because we love garages in Silicon Valley. They're developing psoriasis drugs for Allergan. So until last month, Allergan was going to merge with Pfizer and be the largest pharmaceutical company in the world. Now this team of 7 people full-time is developing drugs for Allergan, the behemoth Allergan, actually. It just takes 7 people to actually do this, enabled with a shitload of data, right? That's the idea here. That's NewMedi, and they're hiring data scientists right now too. All right, where are we going next in the last 4 minutes? What's the next big open data? I can't spend a lot of time on this, but it's clinical trials data. The whole world is heading towards this next big thing. I'm skating to where the puck is going. It's here. Because FDA is going to make the pharmaceutical companies share raw clinical trials data. It's going to be an amazing treasure trove. You're going to look at failed trials, successful trials. I'm showing you the URL here because I run this one for NIH. We're the only website where we give out more than 100 raw clinical trials data. Every patient, every drug, every arm, every encounter, de-identified. You can't figure out who these people are, but you get the idea of what a trials data set looks like. Learn now, because I see where it's coming. Half my research group is just on clinical trials data now, and you can sign up at this for free account here and learn how to do it. Now, I'm lucky because I got recruited to UCSF from Stanford a year ago. We're building this Institute for Computational Health Sciences. We're kind of slumming it out in this brand new building until they build our brand new building right next to where Illumina is, the sequencing company. So you couldn't even buy a better location than that. But of course, the Warrior Stadium will be right next door, so maybe we'll even get some decent restaurants down in Mission Bay, because right now it's kind of a restaurant desert if you've ever driven by there. And that's kind of cool, but why did I move from Stanford? I love Stanford, but why did I move to University of California? Is because UCSF is one of 5 University of California medical schools, right? There's UCSF and UCLA. Those 2 are behemoths. You got Irvine, Davis, and San Diego. And they actually put me in charge of organizing all 14 million patients that get any care in the entire University of California system. That's 4% of the US population gets some care in the University of California system. I'm putting all that raw electronic health record data together in one database. By the way, we're already done. I wanted this done before my first year, so it's living in FSMA compliance over in the San Diego Supercomputer Center on one big Dell box running SQL Server. It pains me it's running SQL Server, but it had to be that. And we have about, for example, nearly 1 billion lab test measurements on these 14 million patients. That's how much data we have. We imagine all sorts of amazing uses of this data. We're going to enable not just us, but enable the entrepreneurs, enable the activists, the social researchers, the cancer genetics researchers, the chief medical officers, the app designers. We have a mandate in the state to really enable everyone, not just ourselves here with this data. We've got to slice and dice and de-identify it. Of course, keep it safe. We've got to keep it private. I'm going to build maps with it, kind of like the last speaker. I love building maps of death and disease. That's kind of morbid, but this is what I mean. Like what do we predict is going to happen next with our patients? So here you see a map like alcohol, mental disorders, that's alcoholism. And then a year later, some come in with liver cirrhosis. And then a year later, a bunch come in with liver abscesses. And then the squares mean they died of that disease. So you can see some people go straight, some people make a detour. They get disease, they get disease, they get death. So I'm kind of figuring out death and disease. Here's a harder one, heart attack. A lot of patients with heart attack die. Some get heart failure, lung disease, and some die of sepsis, which is a bacterial disease. I never knew that happened after 3 years of having a heart attack. And what am I trying to do? This is work by Hannah and Jay. You can see their names there. In our brand new building in Mission Bay, we're going to have a wall of monitors there, and we're going to show our maps of death and disease. But better than just showing the maps, I'm going to visualize all 14 million patients in the University of California system here. This is a prototype of every single patient going from disease to disease to disease to death. And we're going to track all 14 million of our patients. We're going to predict what's going to happen in the next 90 days. We're going to predict what's going to happen in the next year, and we're going to do something about it to prevent that mortality and morbidity. And that's what we're building in the University of California system. I have to thank a lot of people who helped me with the work, the data warehouse team, a lot of collaborators, and a lot of support from UCSF and NIH to do all this work. So thank you very much.